TCA cycle. Today we are going to start about the TCA cycle, that is a tricarboxylic acid cycle, and it was discovered by a scientist whose name was Hans Krebs, and it is also named on the name of scientist who discovered it as Krebs cycle, as well as its third name is citric acid cycle. And what is the TCA cycle? It is a series of chemical reactions that is involved for the formation of energy as well as the energy carrier molecules or the electron carrier molecules and the CO2 that is exhaled through the breathing process. And we know very well that about the cellular respiration. I have already told you that cellular respiration is the respiration or the breakdown of the glucose molecule to produce the energy, as well as the CO2, that the complete oxidation of the glucose molecule. And in glycolysis, I have explained to you that glycolysis is a incomplete oxidation, which means the glucose molecule is half or partially oxidized and converted into the pyruvate molecule. And what next about the pyruvate? That pyruvate is changes into the acetyl-CoA to get entry to inside the mitochondria, because we know very well that glycolysis, which is the first chemical reaction pathway of the cellular respiration, which is the first pathway of the cellular respiration, and from the glycolysis what we have pyruvate, and this pyruvate has to be converted itself into the acetyl-CoA. So it will be easy for this acetyl-CoA molecule to get enter into the mitochondria, because we know very well that TCA cycle as well as electron transport chain, both these pathways, they take place in mitochondria. And if we talk about the structure of the mitochondria, we will know very well that in mitochondria we have an extracellular membrane, or the not extracellular but the extra mitochondrial membrane, as well as the inner mitochondrial membrane. And pyruvate, when this pyruvate has to get enter into the this matrix or inside the mitochondria, it has to convert itself into the acetyl-CoA molecule, and after the conversion of itself into the acetyl-CoA molecule, this acetyl-CoA molecule will come into the matrix, and all the enzymes for the Krebs cycle, they are present inside the matrix. And inside the matrix what we have, all the enzymes which are responsible for the TCA cycle or the TCA pathway. And the enzymes complexes or the enzymes for the electron transport chain, they are present in the inner mitochondrial membrane that we name as crestae. So this is the introduction of the TCA cycle, and this TCA cycle is very important pathway. Like how it is very important, this TCA cycle, it is an amphibolic pathway. What does it mean? Amphibolic pathway means this TCA cycle is involved in anabolic reactions as well as in catabolic reactions. So that's why this pathway is very important, and as well as we also name its reactions or the TCA cycle as the anaplerotic reactions. Anaplerotic reactions, which means it has a replenishing mechanism. What is the replenishing mechanism, or what does it mean? It means whenever its intermediates, they are going to low in their quantity or in the amount, it can fulfill its deficiency by the other metabolic pathways. So that's why we name its chemical reactions as the anaplerotic reactions. So once again I am going to repeat all this. That Tca cycle, it was discovered by the Hans Krebs, so it is also named as Krebs cycle. And the third name is the citric acid cycle. And this Tca cycle, it takes place in the mitochondria, and it is the third chemical pathway of the cellular respiration. And what is the cellular respiration? That is the complete oxidation of glucose into the CO2 as well as the energy. And it involves glycolysis, which takes place in the cytosol, in the cytoplasm, then the pyruvate to acetyl CoA, its conversion. And why it is important? It is important to enter the acetyl CoA into the mitochondrial matrix. And from here, the Tca cycle starts whenever this acetyl CoA is inside the mitochondrial matrix. There starts the Tca cycle, and after that we have the energy as well as electron carriers, and those electron carriers are shuttled to the electron transport chain to get the energy, to get the currency of the ATP, which currency of the energy that we name as ATP. So next, I have explained you that CAC cycle is very significant because it is an amphibolic pathway. What does it mean? It means its intermediate are involved in anabolic pathways as well as in catabolic pathways. And its chemical reactions, they are also named as anaplerotic reactions. What does it mean? Anaplerotic reactions mean they have a replenishing system, which means whenever any intermediate, if any intermediate is low, they can replenish itself by the other chemical compounds, by the other metabolic pathways. So that's why they are named as anaplerotic reaction. And let's start the cycle. What we have is acetyl CoA, and inside the in mitochondrial matrix, what we have is oxaloacetic acid molecule. This oxaloacetic acid molecule, that will, with the help of this acetyl CoA, that will be converted into citrate. And the enzyme involved over here is number 1, citrate synthase, and the second one what we have is the removal of a carbon in the form of CO2 molecule, as well as the removal of CoA-SH from here. And this CO2 the removal of carbon is from the compound oxaloacetic acid, not from the acetyl CoA. And the enzyme involved is citrate synthase, and this is the irreversible chemical reaction. Next one is from here, through two reactions, this will be converted into isocitrate, and the enzyme involved over here is, this is the reversible reaction, and the enzyme involved here is aconitase. This reaction is very significant, very important. Clinically, it's very important. I will explain you all the clinical significance or clinical manifestations at the end of this pathway of all these reactions. About each reaction, I will explain you its clinical significance and what sort of problems we can have. From here, isocitrate, that will be converted into another compound which we name as alpha-ketoglutarate, and the enzyme involved over here is isocitrate dehydrogenase. Remember one thing, whenever you see or you read that in a chemical reaction dehydrogenase is involved, it means there will always be the formation of NAD plus or NADH2 molecule, and here we will have the formation of from NAD plus to NADH or plus H plus. It's also called as NADH2, and as well as we will have a removal of carbon molecule that we name as CO2. From here, this alpha-ketoglutarate, it will be converted into the another compound that we name as succinyl CoA, and here will be the addition of CoASH, and the enzyme involved here is alpha-ketoglutarate dehydrogenase. And as I have told you that whenever there is involvement of dehydrogenase, there will be the formation of NAD plus or NADH2, and in this case we will have NADH plus H positive from NAD plus. This enzyme is very significant, very important. Like the previous pathway, from pyruvate to acetyl CoA, we need a multi-enzyme complex which we name as pyruvate dehydrogenase complex, and that multi-enzyme complex consists of 3 enzymes, E1, E2, E3, and 5 coenzymes, which are the different coenzymes like magnesium, like lipoic acid, like thymine pyrophosphate, and as well as the NAD positive plus and FAD positive. Same here, this alpha-ketoglutarate dehydrogenase is also a multi-enzyme complex, and it also has 3 sub-enzymes or 3 subunits, E1, E2, E3, and 5 coenzymes, magnesium, thymine pyrophosphate, lipoic acid, CoA, FAD positive, and NAD positive. These are the 5 to 6 coenzymes which are involved in this in the multi-enzyme complex that we are naming as alpha-ketoglutarate dehydrogenase. And this enzyme is responsible for the conversion of alpha-ketoglutarate to succinyl CoA. And from here, there will be the removal of this CoA portion and rearrangements, and it will be converted into succinate, and the enzyme here involved is thiokinase. And here there will be the formation of GTP. Let me write this with blue one. Thiokinase, because it is going to cleave the CoA portion from the succinyl CoA part, CoASH, and there will be the formation of GTP from the GDP. And next one is succinate, that will be converted into fumarate, and here the enzyme involved is succinate dehydrogenase. Again, remember, I have told you that whenever there is the involvement of dehydrogenase enzyme, there will be the formation of any electron carrier, and mostly we have talked about the NAD positive and NADH, H plus, H plus. But when the energy gradient is low, then NADH will not form, but it will form another electron carrier that we name as FADH2 from the FAD positive. So here we will have the formation of FADH2, and again there will be the removal of another and after that from the succinate dehydrogenase, this enzyme is also very significant. Why this enzyme is so significant? The main reason is this is the enzyme which is a complex 2 of electron transport chain, and this is the only enzyme which is present on the inner side of inner mitochondrial membrane. I have told you that all the enzymes that they are present in the mitochondrial matrix, but this is the only one enzyme that is not present in the mitochondrial matrix, but it is attached to the inner side of the inner mitochondrial membrane, and it is also the complex 2 of electron transport chain. And from here, from the fumarate to what we will have another enzyme, the malate and the enzyme involved is the fumarate. And then again there will be the dehydrogenation, and with the help of dehydrogenation, the enzyme involved is malate dehydrogenase. As I have already told you that whenever there is involvement of dehydrogenase enzyme, there will be the formation of NAD positive or the NADH, which is electron carrier like this. NAD positive, that will be converted into, and again we have oxaloacetic acid, and it will start a new cycle, a new Krebs cycle. So these are all the chemical reactions which are involved in the TCA cycle. Now remember, let me tell you about the irreversible and reversible pathways. This reaction is reversible. Then all these three reactions, these are irreversible, and from here this reaction is also reversible. All these three reactions are irreversible, and this one is reversible. This is also reversible, and this reaction is also reversible. So, reaction number 1 and this one. These 4 reactions, these are irreversible, while all the remaining reactions are reversible. And these are the chemical reactions of the TCA cycle or the citric acid cycle that we have studied, that I have explained it to you. Let me once again repeat this to you. This acetyl CoA that will be attached with the oxaloacetic acid, and there will be the removal of CO2 as well as CoA-SH, and the enzyme involved is citrate synthase, and what we have the product, that is the citrate. Citrate, with the help of a conidase enzyme, that will be converted into the isocitrate, and this isocitrate compound, with the help of dehydrogenase enzyme, it will be converted into the alpha-ketoglutarate. And whenever I have told you, whenever there is involvement of dehydrogenase, there will be the formation of electron carrier. Here you can see NADH plus H positive, and as well as there will be the removal of CO2 molecule. And next one is alpha-ketoglutarate, that will be converted into the succinyl CoA, and the enzyme involved over here is alpha-ketoglutarate dehydrogenase, and again there will also be the removal of CO2. And from here I have told you that this enzyme is same as like the pyruvate dehydrogenase multi-enzyme complex. It has 3 sub-enzymes and 5 coenzymes that I have already explained to you. And from here alpha-ketoglutarate to succinyl CoA, what we have? Succinyl CoA. This succinyl CoA, with the help of thiokinase, it will be converted into another form, succinate, in which we will have the rearrangement of the atoms in the succinyl CoA as well as a cleavage of CoA portion. And here the energy gradient will be too low, and we will have GDP in the form of GTP. We can say that this GTP is converted into the GTP, and this GTP that will be converted into GDP, and it will give the ATP with the help of dinucleotide dikinase enzyme. With the help of kinase enzyme, it will remove one phosphate from here, and it will attach this phosphate to the ADP, and what we will have? We have ATP, and once again our this molecule is ready to form another molecule of GTP. So from here the succinate, it will be converted into the fumarate with the help of enzyme succinate dehydrogenase, and I have told you that this enzyme is very significant because this is not in the mitochondrial matrix, but it is present on the inner side of the mitochondrial membrane, and it is a part of the second complex of the electron transport chain. And from here what we have at the end, we will have at the end a fumarate molecule, and this fumarate molecule will be converted into the malate with the help of an enzyme that we name as fumarase enzyme. And this malate, it will be converted into again oxaloacetic acid with the help of malate dehydrogenase enzyme, and again there is involvement of dehydrogenase enzyme, and what we will have at the end, NADH plus H positive. So oxaloacetic acid is ready to start another Krebs cycle. So these are all the chemical reactions and as well as the enzymes of this TCA cycle. Now the next thing is how you can remember this. It's very easy. Let me give you a mnemonic to memorize this. I'm going to erase this. And what is that mnemonic? It's very simple. Sorry. Citrate is a specific substrate for making oxaloacetate. What is the mnemonic? Citrate is a specific substrate for making oxaloacetate. How? Citrate, here we know very well, this is a citrate. Then is isocitrate, you can see here, isocitrate, citrate is. Then it means alpha-ketoglutarate. And the next one is the specific means succinyl CoA. The next one is the substrate, it means the succinate, and for that stands for fumarate, and making that stands for malate, and oxaloacetate you know very well. So its mnemonic can be citrate is a specific substrate for making oxaloacetic acid or oxaloacetate. So you can memorize the chemical reactions with the help of this mnemonic. Citrate is a specific substrate for making oxaloacetic acetate. And the next thing is which students the mostly suffer, to memorize the name of the enzymes. Remember about the reaction, about the substrate which is going to involve in that chemical reaction. All names of the enzymes, they starts from the substrate and end with the chemical reaction, with the type of chemical reaction that is going to happen or going to be within that chemical reaction. For example, isocitrate, it's going to convert itself into the alpha-ketoglutarate. So how you will name it as? We know very well that this type of chemical reaction is a dehydrogenation reaction. So how we name it as? Isocitrate dehydrogenase. I have told you that first of all, pick the name of substrate, then at the end add the type of chemical reaction with ending ase, like isocitrate dehydrogenase. Type of chemical reaction is dehydrogenation, substrate is isocitrate, like this. What is the substrate? That is isocitrate. And what is the type of chemical reaction? That is dehydrogenation. So how you will name it as? Isocitrate, and end the name of chemical reaction with ase, like isocitrate dehydrogenase. So like this you can memorize the name of enzymes in all chemical reactions or of all chemical reactions. I think this is clear to you. Now the next thing is, what is the end product of this process? What we have at the end? At the end we have, if we calculate its energy budget, at the end what we have from here, let's start here, one NADH, here second NADH, here third NADH. So at the end we have 3 molecules of NADH2. And we know very well that in the glycolysis we had 2 molecules of pyruvate, and if those 2 molecules of pyruvate, they will enter into the PDH complex pathway, then we will have 2 molecules of acetyl CoA. Same here, we will have 2 molecules of acetyl CoA. It means this Krebs cycle will operate twice, not once. So we can say that, then we can multiply it as by 2, and what we have, 6 NADH2. And again here we have FADH2, 1 molecule of FADH2, and this cycle is operating twice. So we will multiply it with 2, and at the end we have 2 FADH2 and 1 molecule of GTP, and we know very well that this reaction is operating twice. So we will have 2 GTP which is equivalent to 2 molecules of ATP. So if we calculate it at the end, we know very well that 1 molecule of NADH2 is equal to 3 molecules of ATP, like this. 1 molecule of NADH2 is equal to 3 ATPs. Actually it is 2.5 ATPs, but we write it as 3 ATPs. In most books you will find 3 ATP, and in few books you will find it as 2.5 ATPs. And how many NADH2 we have? We have 6 NADH2. So if 1 NADH2 is equal to 3 ATP, then 6 NADH2, that would be equal to 6 multiplied by 3, and it will be 18 ATPs. So 18 ATPs are here, and 1 FADH is equal to 2 here we have 2 FADH2 molecule, and 1 FADH2 molecule, it is equal to 2 molecules of ATP. I will prove this, that how 1 NADH2 is equal to 3 ATP and how 1 FADH2 is equal to 2 ATP. I will prove this in electron transport chain. And how many FADH2 we have? We have 2 FADH2. So 2 FADH2, that will be equal to 2 multiplied by 2, that is 4 ATP. And how many GTPs we have? We have 2 GTPs, and we know very well that these 2 GTPs are equal to 2 ATPs. So let's calculate it. Number 1 from NADH2, number 2 from FADH2, and number 3 from GTP. So let's calculate this portion. From the NADH2 we have total number of ATPs, that is 18. And from the FADH2 we have total 4 ATPs, and from GTP we have total 2 ATPs. So let's calculate it or add it. 18 plus 4, 22, 22, 2 plus 2, that is 24 ATPs. So in the Krebs cycle we have total 24 ATPs if this cycle operates twice, and if it's operate once, then we have 12 ATPs, but when it operates twice we have 24 ATPs, because we know very well it will always operate twice, because we know very well that we have 2 molecules of acetyl CoA from the coming pyruvate, and so it will operate twice and it will give 24 ATPs total at the end of this pathway. I think this is clear to you. And in glycolysis we know very well that in glycolysis we had 6 molecules of ATP, and glycolysis plus this one Krebs cycle we have 30 ATPs, and in PDH2 acetyl CoA we also have 6 molecules of ATP, and 30 plus 6, the total budget will be 36. Let me show you how it is 36. I am going to raise this. In glycolysis, we have 6 ATPs. In PDH, complex pathway, we have 6 ATPs, and in TCA cycle, we have 24 ATPs, and calculated it will be 36 ATPs. And so we can say that with the complete oxidation of 1 molecule of glucose or 1 gram of glucose, we have 36 molecules of ATP. So 1 molecule of glucose, that is yielding 36 molecules of ATPs. That is quite enough for a cell to perform its activities. I think this is clear to you about all these chemical reactions. And the next one is CO2, 1, 2, and here 3. So in this we have 3 molecules of CO2, and we know very well that this TCA cycle is operating twice, so we will have 6 molecules of CO2. You know very well about the equation of the respiration, C6H12O6 plus O2, that will be converted into 6 CO2 plus 6 H2O. So here 1, 2, 3, and this cycle is operating twice, so in total we will have 6 molecules of CO2 in the Krebs cycle. Is that clear? So what is the end? 6 molecules of CO2 and 24 molecules of ATPs. That is the end of TCA cycle. I think this is clear to you.